Hi, my name is Leonardo Cortez. I'm a research associate in Dr. Valerie Sim's lab at the Center for Prion and Protein Folding Diseases in University of Alberta. So first of all, I want to take this opportunity to thanks very sincerely to the CJD Foundation and especially to the donors that decided to support our research. Today, I want to give you a brief overview of this project that focuses on studying how different types of prions drive different disease phenotypes. We know that prion diseases can be presented with a broad and variable range of symptoms. We also know that during prion disease, the normal prion protein misfold, change its shape, and start clamping together to form a wide variety of prion particles. Prion with different shapes and different properties. We can divide these prion particles in two groups, two populations. One that include the smallest particles, the particles that are less stable and possibly more toxic. And a second population that include the larger particles, more stable, much harder to be degraded by the brain. These two populations of prion particles also differ in the ability to replicate and spread in the brain. So in this context, a natural question that comes to our minds is there any association between these two populations of prion particles and the different symptoms that a patient with CJD present? Well, in an article that we recently published, also with the support of the CJD Foundation, we found that in fact there is an association. We proposed that the different proportion of these two populations of prion particles in the brain of patients with CJD will drive their symptoms. However, there is a very relevant aspect that we haven't explored yet, an aspect that will help us to know more deeply about this association between type of prion particles and symptoms. And that aspect is the regionality. With regionality, what I mean is figuring out how these two populations of prion particles spread and how they grow in different regions of the brain. So each brain region is a specific, a very unique environment that can favor one particular population or one particular type of prion. Then the consequence of that is that we will have different degrees of damage in different regions of the brain, with the consequent different symptoms for that disease. For example, if more neurotoxic prion particles accumulate in the occipital region of the brain, that patient will have vision problems. So in this project, using the same techniques and same method that we used before, we will try to isolate and characterize the different type of prions accumulated in different parts of the brain of patients with CJD. This will allow us to understand better why different patients have different symptoms. And ultimately, this knowledge could help us to better target specific type of prion based on the symptoms of the patient. Another interesting question that we will try to answer is which factors control the propagation of these two populations in the brain of a patient with CJD. We know that the prions are formed in the membrane of the neurons, and the membrane is a particular structure that is composed mostly for lipids, a specific type of molecule that is present in all these membranes. So what we will try to do is try to identify which lipids are present in each region of the brain and try to associate the type of lipid in that region with the type of prion that is accumulated in that specific region. If we identify which lipid influence the propagation of specific type of prion, then we will have a new target toward which to direct our therapeutic strategies. So those are our goals, and with your support, we will try our best to get there. So thank you very much again. Thank you especially to the donors. And hopefully next time I will be able to show you exciting results.